Webinar today is particularly important because of for a few reasons. One, COVID is already endemic, probably. Two, we have the Omicron surge. And three, we have large numbers of the population being vaccinated. And these are dynamics that will change the way we manage COVID. So into this picture, we will get to discuss today the cPass test for neutralizing antibodies. It is a blood test using the ELISA technology, and it is produced by a company called GenScript, which is one of the three companies that are organizing today's webinar. GenScript, the manufacturer, Biomed Global, the importer distributor in Malaysia, and Gribbles, the laboratory in Malaysia that will be managing these tests for you. So Professor Chan and Dr. Leong, the audience today, so far we've got about 180 participants and more will be coming in, and there will be general practitioners and family physicians in the private sector in Malaysia largely, and there will be a few from the public sector. May I invite Professor Chan to perhaps begin her comments, and please feel free to share your screen, Professor Chan, when you will be talking to us a little bit more about the science of neutralizing antibodies. Professor Chan, it's a pleasure to have you. The floor is yours, and very happy to see your screen right now. The floor is yours.
Can you hear me?
Yes, we can, Prof. Chan.
Yeah. Okay. So thank you, SK, for the invitation, and I'm glad I can share some of my views on neutralization antibodies. So today I'm going to provide some basic understanding of what is neutralization antibody. So so basically, my slides will consist of the some explanation of what is antibody, what is exactly neutralization antibody, and is this actually important for vaccines? So let's recap back a bit of our understanding about immunity. So we know that immune responses is actually a defense against pathogens, and these pathogens that we are referring to actually could be virus, could be bacteria, could be parasites, or could be fungi. So, as we understand, there are two arms of the immunity: the innate and wait, let me just get my pointers out. So we have innate immunity and adaptive immunity, and what exactly are innate immunity? So innate immunity is basically some of the characteristics is actually non-specific. It usually when you actually encounter a pathogen, your body actually quickly responds to it. And the examples of components of innate immunity are examples are like our skin, our mucous membrane, the cells that are listed here like macrophage, neutrophils, dendritic cells, eosinophil, basophils, and also cytokines and complements. So on the other hand, that's another arm of adaptive immunity, which is usually known as specific, and this takes longer to actually produce and to actually to protect against pathogens. So we actually over here we have something called the humoral antibody, humoral or antibody response, and also the T cells or cellular response. So both this innate and adaptive immunity can help us human to protect against the pathogens. So so if you look at this particular busy slide, but I will walk you through how our body, our immune response occur after we have COVID-19 vaccination. So basically what happened is we have one dose of vaccine, and we know very well, okay, this illustrates against the spike protein, which is the subunit vaccine, but it could also be inactivated vaccine. And with this, you know, all these proteins of the vaccine will then enter the cells. And then what will happen specifically, it's your helper T cells that detect the antigen, which could be spike or could be any inactivated parts of inactive vaccine. It will be other parts of the proteins that will be actually presented by helper T cells to alert other immune cells, and then you will have killer T cells to actually sit out for this antigen, or your B cells will actually create antibodies that recognize the spike proteins or any other proteins of the vaccine. So with this, your T cells and B cells will develop immune memory, and in future, when you encounter the virus, you will actually have a response. So we know that antibodies remain in your body for some time, and this antibody is specific against, if it's made for spike, it will be specific for spike. So with that, after second dose or after subsequent booster, we would actually train our immune system to actually be able to recognize, to actually produce more antibody and have memory against the virus. The slide is not moving. Okay, so what is then we know about antibody as well as T cell response, but what exactly is neutralization antibody? So if you look at this diagram in laboratory, we do this particular test called plaque reduction neutralization test, which is a gold standard to actually test neutralization antibody. So this neutralization, if you do plaque reduction neutralization test, what happens is that it's very laborious. It requires live virus, and it can only be done in certain very limited laboratory. So therefore, that's a restriction to do such a test. But clever scientists have actually used a certain way to actually not to do plaque reduction using live virus, but they have actually used a certain ways. For example, use this particular assay called using receptor binding domain of a spike, how it actually binds to the ACE2 receptor, which is the actually receptor for SARS-CoV-2 in the whole cell. And what you do is you block this complex, and that will actually measure the neutralization antibody. So basically, neutralization antibody is actually it binds specifically to antigen, if in the case it's a spike, and specifically it's the receptor binding domain. So what it does is actually prevent virus from interacting with the cells. But however, when you actually have when you get if your body produces antibody, whether through natural infection or through vaccine, it can still bind to the virus, but it will not affect the infectivity. Unlike neutralization antibody, what neutralization antibody does is it will bind, it will also actually prevent infection. So you can see from this diagram here, basically, if you actually measure the binding when it's negative, you will actually be able to measure the inhibition. And if you can actually see here, when there's actually antibody in the body, it will prevent virus from actually binding, and infection is prevented. So I would also like to introduce to you a term called correlates of protection. So basically, it's actually measuring vaccine or natural infection immune response, and how it actually can protect you from the same viral infection in the future. So basically, we could use antibody, or we could use actually T cells to do that. And basically, if you measure antibody, it's actually measuring how it binds to the virus, how subsequently the binding correlates with actually no infection and protection. So in T cells, it will be measuring actually how the T cells actually recognize and destroy the infected cells. So they are actually growing evidence of how important is neutralization antibody, especially for a lot of vaccines, and also recently for COVID vaccine. So this is a paper by Cowrie and colleagues, which shows that neutralization antibody levels are highly predictive of immune protection from symptomatic SARS-CoV-2 infection. So there's also another paper by a very famous professor called Plotkin, Stanley Plotkin. It's basically a guru of correlates of protection, and he believed that antibody is at the moment the best marker to actually correlate of protection against for COVID vaccines. So the other paper that came out recently, so the other paper that came out recently is this particular paper in Lancet by Cromer and colleagues, which actually states, which combined about 24 papers of to do this meta-analysis and shows that neutralization antibody is actually likely to be a predictor of protection against SARS-CoV-2 variants, and this actually shows that, you know, if you actually have a booster, that will increase the neutralization antibody titer against variants. And last, of course, this is the interesting, as we have growing concern about Omicron. So Pfizer has recently showed that if people who take 2 doses of vaccines will have limited antibody levels against the Omicron, but if you actually have a booster, you will actually 5 times more protection with the higher antibody level. So so I hope with this short presentation, I have actually showed you, you know, what exactly we know about neutralization antibody of what exactly is neutralization antibody, and actually what we know about neutralization antibody in SARS-CoV-2. With that, I pass back the time to SK.
Thank you, Professor Chan. I think that there were 2 important points that maybe we can surface during the Q&A session. And for all those who have just joined us, welcome to the webinar that we're hosting. When I say we, what I mean is GenScript, the manufacturer of cPass, Biomed, the importer distributor of cPass in Malaysia, and thirdly, Gribbles, the laboratory that is offering cPass for you, the general practitioner in Malaysia. cPass is a blood test for neutralizing antibodies that can be offered to people who have been vaccinated or are considering getting their boosters. Ladies and gentlemen, what you just heard was who you just heard rather was Professor Chan Yoke Fun, medical microbiologist and virologist from University Malaya. She says that there'll be 2 important things that I take away. Not all antibodies are neutralizing because binding antibodies do not interfere with the infectivity of COVID. So not all antibodies are neutralizing. And secondly, neutralizing antibodies are increasingly being seen as approximating the correlate of protection. Thank you very much, Prof. Chan, for your comments. So from the science perspective of neutralizing antibodies, let's now turn to Dr. Leong Hoe Nam, who is an infectious diseases consultant in various private hospitals and clinics in Singapore. And Dr. Leong is a very accomplished clinician, and he will talk to us for about 12 to 15 minutes about the clinical uses of cPass, as well as how to interpret the results. Dr. Leong, the floor is yours, and I'm told that you'd like to share some slides. Dr. Leong, very happy to have you here. The floor is yours.
Yes, thank you. Thank you. SK, I really like you. I like you very, very much. You're really right to the point. Now, Malaysia likes to look at Singapore politics. So if there's anything you might know of someone called Paul Tambai, who's also an infectious disease physician, and he came up with this slogan, ask Paul anything for his elections. Unfortunately, he didn't make it for the elections. I was kind of rooting for him. But more importantly, I like his tagline, ask anything. Ask Paul anything. But in this case, I'm going to ask you to ask us anything. Professor Chan or myself or GenScript. I promise you we'll only share the truth and nothing but the whole truth. Okay, I'm going to go on to the talk now, and I'm going to talk a little bit about serology testing. I know Professor Chan covered about neutralizing and non-neutralizing, but I'm going to show it in a slightly different way, which you can try to replicate for your patients when they're in front of you, about the different tests there are. I'm going to talk a little bit about booster vaccines, why we're doing it, and how they measure it. In a way, it is similar to Professor Chan's talk, but a little bit of a different slant. And then something called hybrid immunity, which I'm really rooting for, and what happens in a clinical setting. So when you actually do the antibody test, which you have done for the chickenpox and the measles, we ask for total antibodies, total measles IgG, total measles VZV, and with that, you determine if the person has been infected or not. So this neutralizing concept is rather new. Neutralizing, that means it kind of blocks off and negates the effect of the virus. The virus becomes non-effective. That's what you like. You just don't want, you don't want the antibody to bind to the virus like appendage that's not doing anything. You want it to stop it at its tracks. And the way to have it is with neutralizing antibodies. If you look at the spike protein that's down there, you realize their antibodies can bind anywhere. But the one particular one, in this case, the black one, would interfere with the binding of it. It's like a stopper at the door. You have a stopper at the door and the door can't close properly. And because of that, the spike protein can't bind into the receptor as a blockage. With this blockage, you block the binding. You block the binding, the virus doesn't enter the cell, we effectively neutralize the antibodies. So that's what I like about it, because if you think about it, it's almost not exactly like the quality test for the antibodies. If I have all the antibodies are all blocking, then I effectively can prevent COVID-19 infections. So some of the test kits offered by Roche, Abbott, and Siemens, they do the total antibodies test and they measure all the black and the red ones. But if you actually do the cPass, they have a neutralizing antibodies, it is actually the black one. So you actually measure, you truly measure the antibody that blocks the binding of the virus to the cell receptor, and that's the one you want because you effectively block the virus from entering. So we talked a little bit about this. Professor Chan talked about Pfizer, why we do boosting, and this is a similar study, but this is from Israel. Israel did their studies and then they did it and they realized that among the various age groups, the risk of confirmed infections, I want to stress to you, if confirmed infections is not hospitalization, comes down with boosting. And what happens is, if you look at the severe cases and non-severe cases, you actually see a reduction in numbers. The rate ratio reduction comes down to about 18.7 to 22. You see a nice fall in numbers. Great. So I like it because we have fewer infections and fewer severe disease. But if you look at the 16 to 39, the numbers which you can reduce, well, it's too few. You can't really quantify it accurately. But very importantly, booster doses help. But what does booster dose do? It actually rings up your antibodies. Number one, if you do the timing correctly, your antibodies will mature inside the marrow, or rather the B cells will mature inside the marrow, resulting in more potent antibodies. Number two, you will also train your T cells at the same time. So not only will you have your B cells that are strong and your T cells that are strong, and Professor Chan alluded to it earlier on. So coming back, booster dose does work. And when you actually do boosting, you can do it with the same vaccine like homologous, you can do heterologous, which is slightly different, or you can do hybrid boosting, which is infection plus vaccine. But when you do hybrid boosting, something happens. I'm just going to show you this slide very quickly. It's about booster vaccinations with heterologous boosting. That means one moment is Pfizer, next moment is Moderna, and next moment is Janssen and Janssen. So when you actually use these three different boostings, what are the effects? And how did they measure all this? With the antibodies. I'm going to show this to you now. On the y-axis, you have the anti-S antibodies. So the high levels you have, then the greater the anti-S antibodies you have. But the graph on the lower actually measures the neutralizing antibodies. Again, the higher you are on the y-axis, the more neutralizing antibodies you have. And you notice, in this case, I put in this message, cPass measures this. cPass is a measure of the neutralizing antibodies, and it tells me quite confidently the quality of antibodies I'm getting and how much of it is being blocked. So in the primary series, you have people who had the Janssen and Janssen, mRNA, as well, mRNA representing Moderna, and of course Pfizer. So you have the Moderna boost, you have the Janssen boost, and finally you have the Pfizer BioNTech boost. What happens to this boosting? Let's look at the antibody levels, and we're going to show it to you here. This is it. Moderna boosts very, very well. Sorry, Janssen and Janssen. If you do a Janssen followed by Janssen boost, then the amount of antibodies which you have is very, very little. And this will actually translate into clinical results saying that it doesn't work so well. If on the other hand, you had a Moderna, followed by Moderna, followed by Moderna, you actually have a very good boosting. And the last case is actually that of the Pfizer. You have Pfizer followed by Moderna boost. It's actually quite good. So the summary slide is this. If you look at the antibodies, which is a good marker, looking at the neutralizing abilities, the Janssen and Janssen boost is the worst. It's the worst. So you realize the makers of the vaccine are quite smart. They only designed it for one shot, but they didn't plan for COVID-19 or coronavirus to keep adapting and mutating such that you need booster shots. Moderna boost is the best. If I can have, I want to have a MMM. If I can't, I want to have a PPM. And that's the third point. Pfizer followed by Moderna boost is actually very, very good. Now, so we talked about antibodies, we talked about boostings, but there's a special scenario when you actually do a post-COVID infection and vaccination. So there are people who are, oops, sorry, this is not supposed to show up. Okay, there you go. I wanted to go up to this side. So what happens is when you have individuals who have recovered from a strain, say Delta or Omicron, for example, and thereafter, when you give a COVID vaccine, what happens to them? I'm going to show this to you. You notice there are two colors, the white ones with the open circle, as well as the red one. What happens is, on the left of the white, after having an infection, you realize that the antibodies against the different strains aren't very good. You can't really cross the line, the horizontal line, which is a marker of the immunity. But when this group, which are all white circles, they go on for one vaccination, they become red. When they become red, you notice that all the immunity, which is the neutralizing antibodies, are all above the line, indicating that they can deal with the variants of today and variants of tomorrow. If you look here right at the bottom, there's a PCO on the right-hand side, PCOV GX and BCOV WIV16 and SARS-CoV. You notice these are the variants that are still in the caves in Yunnan or Thailand or Cambodia as you find them. And you notice SARS-CoV, there's SARS-CoV-1, which is SARS virus of 2003. So they are able to cope with them when you actually have vaccination, infection plus vaccination. So together you have usually what you call the hybrid immunization. And this is the line which I'm going to repeat in verbatim for you. Plasma from individuals who have been infected and subsequently received the mRNA vaccination. They do, oops, what happened? They actually neutralize, including those pseudotypes. Pseudotypes, that means they didn't use the actual virus, but the other spike proteins which represent them, and they overcome a diverse Sarbeco virus, which means the variants of today and the variants of tomorrow. Now, this is a point which I'm going to stress again, which Professor Chan talked about. The immunity of the individual actually has two arms. One is a B cell arm and T cell arm. And when we do vaccination, we are actually trying to stimulate both of them. But I'm going to look at it from a different perspective. And I call this on the ground perspective, the clinical perspective. What does it mean to the common person on the ground? If I have antibodies that are measurable, it tells me that I have enough antibodies to neutralize it. It's the first line of defense. But if your first line of defense breaks through, what helps you next is actually the T cells. The T cells is your second line of defense, and this is where it determines the severity of illness. On the case of the B cell arm, because it changes, the antibody kind of binds to the spike protein. If the spike protein changes, it gets affected by mutation. But in the case of the T cell arm, the mutation is less affects the T cell arm of it. In other words, it may be alpha, beta, gamma, delta, Omicron. The T cell can kind of still remember it, which is great because the variants will occur. You break through your B cells, that means you have an infection. but because you have T cell, you actually end up with less severe illness. So this determines the infection and this determines the severity of illness. So if you were to do a cPass test, you are measuring for the individual what's the chance of the person falling sick. But it doesn't tell me what's the severity of illness the person is going to suffer. Agree, the higher the antibody levels, the better it is to prevent. And this is what you want. You want to prevent the infection from happening. The next question is how much cPass is needed, how much protection is needed. There is an 80% protection. So if you can cross the 80% protection, you will probably be able to block Delta. I'm going to stress this again. This is for Delta. Now, when I last spoke together with Professor Wang Ningfa, who's the inventor of this test, they actually came up with a dPass. dPass placed on the word Delta, which is a cPass that uses a dPass spike protein. However, with the Omicron, it kind of messes up their entire workflow. The dPass is no longer relevant, but they might need the old pass. But you realize that this becomes the problem of the manufacturers. Is Omicron the last one? If Omicron isn't the last one, it will be a Z plus or something else, or I plus or EHU. It becomes very difficult. When do we stop making? So at this point in time, we're still quite happy with cPass. cPass at 80% refers only for Delta. For the Omicron protection, we don't quite know, but I presume it's going to be 80% plus a little bit more. We're still waiting for data. Now, why is it important to look at cPass for some of these people who have been vaccinated? This is a short series of very skewed data of patients who had their Sinopharm vaccinations at our clinic. And if you look at their cPass levels, after 3 shots, about 2/3 of them would respond with more than 90%. But there are a significant number who have cPass of less than 80%. What it tells you then is that many people will still succumb to the illness and perhaps succumb to the disease. Why? Remember, the adjuvant that's used in the inactivated vaccines, which is in this case Sinovac and Sinopharm, is aluminium. Aluminium is a poor T cell stimulator. So you may have some antibodies, but the illness will fall through, and in turn, you still fall quite sick because of your T cells that are weak. So I talked about T cells quite a few times, and what I really want you to know is cPass, and this I have permission from GenScript, cPass only looks at one arm of B cells. But what you really want to look at is the B cell and the T cell combination. So a measure of T cells, well, there are companies that are trying to do it. On the left is something from Duke and US. They came out with a rapid test. They are trying to actually provide a commercial service for it, but they are still quite far away from that. QIAGEN has an FDA-approved one recently. It's called QIAGEN quantiferon. And you recognize the name quantiferon because it's very similar to the TB quantiferon. You also notice that on the right-hand side of the screen now, you can PerkinElmer, who actually comes out with a T-spot discovery. There's Adaptive Biotechnologies, it's come out with a T-Detect. And of course, you can find on the internet a T-spot COVID, but that has not been approved yet. So at the end of the day, I would really want to measure B cells and T cells, and that will give the full picture about the immunity of the individual. Okay, coming back. As a clinician, what do I do? When do I use the cPass? Well, I look at the immunity of the person. I look at the vaccines the person has been received. If I had 3 times mRNA vaccine, everybody, practically everyone, will have more than 90% antibodies, usually in the region of 96 to 97%. But if the person is immunocompromised, is on CellSAP, prednisolone, etc., I would actually want to do a cPass to measure the amount of antibodies a person has. And the antibody levels are low, I would actually consider going for even a 4th dose vaccine. And this is what the Israelis are doing. They're going for a 4th dose, but only for those people who have a poor immune response. Remember, at this point in time, I don't have a T cell test. Now, I do have many people who are very hesitant to the 3rd booster shot. And every day I'll see 2 to 5 such individuals in my clinic, and I'll tell them you need to get your booster shots, etc. And one of the ways I do it is to actually look at the cPass result and use the 80% quantification, which we talked about. Your cPass, my friend, for example, patient, is only 60-40%, which means you're highly predictive of falling sick with Omicron. In fact, Omicron will probably need something more than 80%. So my dear friend, please go ahead with the booster vaccination. Now, we have many individuals who refuses to touch the mRNA vaccine with a 3-foot pole. So in these individuals, they will want to know what's the immunity. Is it enough? Or do I need another shot, or what's happening? And what's the level of protection? So I do a cPass for them to quantify. And you know that eventually some of them do very well. Those are rare minority where they have about 80 to 90%. But sometimes you end up having to convince them to change their mRNA vaccination instead. I do have a patient who's data-centric. He says, I want to know what's the pre and what's the post. And I say, you have already had 2 doses of mRNA. You are really quite good. Just go for the 3rd shot, save the money. He says, no, I want to see the data. I want to see what is it before and what is it after. So there are people who are very willing to look at the levels going up. They want to see the pre-post effect. So the other thing which I need to stress to you is these antibodies which you do now, today, for example, is just a spot at this point in time. If you give it enough time, the antibodies will fall. However, a small proportion of people, up to 10 to 30% of them, the levels do not fall at all. It remains very, very good for the next 3 to 6 months. So those people who fall, then you actually need your antibodies up. Antibodies up, it becomes your first line of defense and to prevent the infection from falling through. Yet, I want you to remember, the B cells antibodies only tell one end of it, but what you really want to know is actually the T cells. At this point in time, I'm going to highlight show you something from the EU drug regulator. You know the companies, Pfizer and Moderna, they're saying booster, 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 booster. EU has come forward and say, no, perhaps we don't need a booster shot. We need a 3rd booster shot, but perhaps we don't need the 4th booster shot. And they went on to say that if you get repeated boosters, you could actually weaken the immune system. And there's truth in it. If you keep challenging and challenging and challenging, you could end up in a situation where the body says, I'm going to treat this as self, no longer as an enemy. And if I really want to wait, I will want the immunity to mature in the marrow. And the earliest I should do a shot is 3 months, though ideally I like it at 5 to 6 months. And this question point goes back to you, the audience. Do I really need a 4th shot? Do I really need something after the 3rd shot, another booster? Or is there something better? I gave a hint of my views in the earlier slides, and I just want you to think about it. Just another teaser here. There are companies that are coming up with vaccines to actually do the T cell training alone. They focus on stimulating the T cells rather than the T and B cells because they realize that the B cells will fall. And what you really want is T cells to keep the strength of the immunity good. That means changing a serious illness to mild illness or no illness at all. That comes to the end of my slide. I'm going to pass it back to SK. SK, you have the floor, and please remember, ask anything.
Dr. Leong, thank you so much. You've given us a tour of the clinical uses of cPass, specifically when to use it, how to interpret the results, and also how does that relate to vaccines for people who are waiting for the third dose or waiting for boosters and things like that. Thanks very much, Dr. Leong, for all the information that you've shared with us. If you've just joined us, ladies and gentlemen, this is a webinar about the use of cPass and the clinical uses of cPass neutralizing antibody testing in your clinical practice. Earlier, before Dr. Leong, we've got Professor Chan, who spoke about the medical microbiology, the virology, and the scientific aspects of immunity and cPass. And Dr. Leong has provided us an explanation about the clinical uses of cPass. Dr. Leong and Prof. Chan, there are some questions from the floor right now. So maybe we'll take some questions from the floor because there are already some in the chat group. So what I will do is you can ask your questions in 3 ways. Number 1, if you raise your hand, you can verbalize. Number 2, you can type it in the chat group and I'll call some questions from the chat group. Or thirdly, if you prefer not to be identified, you can also send me a private message on Zoom. Ladies and gentlemen, let me start with a question that is a little bit outside of the conversation, but it tries to bring everything together. My first question is to Professor Chan. Prof. Chan, there have been multiple arms of the immunity system that we've talked about. Neutralizing antibodies, binding antibodies, B cells, T cells, CD4, CD8, and natural killer, and so on and so forth. Can you tell us a little bit, what are the relative contributions of neutralizing antibodies and the other arms of immunity as it relates to the overall protection against SARS-CoV-2 or the COVID virus? Professor Chan, over to you.
So it is $1 million question. So I think it's a very complex relationship between all these cells. So basically, I think we are made beautifully by God, and that's what our immune systems are actually a lot of redundancy. And like what I mentioned and what Dr. Leong has mentioned, at the moment, the science that we have actually shows the importance of neutralization antibody, and that is easy to do. That's very easy to be measured, and therefore, as the science progresses, we think for now at the moment the best is actually we find that there's a correlation of protection with neutralization antibody. So that's easily can be done. However, as what has been mentioned by Dr. Leong, T cells are relatively important because people have, we know that despite emergence of many different variants, actually still conserved, and therefore we believe that T cell immunity has not really changed despite the different variants emergence. So with that, T cells would be able to actually protect you regardless of any future or current variants. And therefore, T cells, like what has been mentioned, it could predict, it will predict severity. It can actually, when you do not have antibodies, that doesn't mean that you do not have protection. There are also memory B cells that we do not have easy assays to measure. So at the moment, I would think, if we are allowed, scientists are allowed to measure every single types of, from neutralization antibody to memory B cells to T cells to cytotoxic T cells. I think that's never ending to all the assays. So therefore, at the moment, science only recommends, to mainly work on the easier ones, which is neutralization antibody.
Noted. Thank you, Prof. Chan. Prof. Chan, as a small follow-up question, as we get more questions from the audience, to do a B cell test, is it available in Malaysia right now?
When you want to measure memory B cells, you will need to actually collect blood from a patient, a volunteer, and then subsequently you would need to actually sort out the memory B cells, which is quite difficult and requires a special instrument called flow cytometer. So that could only be done at the moment with research tools, and even with research tools, it can only be done in very specialized laboratory. So at the moment, it can't. It is not a routine test for now. I think there have been studies to show that, despite if you have a vaccine and you do not have high neutralization antibody or high and therefore it could protect you from actually infection or severe disease.
Thank you, Prof. Chan. A B cell test or memory B cell test is available in Malaysia, but it's very complicated using a flow cytometer.
I will refrain. I don't think I should say even available. I don't think it's available. So it's very much a very limited research tool.
That's a good point. Thank you, Prof. Chan. So in summary though, a memory B cell or a B cell test in Malaysia is for all intents and purposes, extremely difficult to get. Very complicated, takes days to achieve a result, and it's not easy. Therefore not cheap. Based on what Dr. Leong has also mentioned, a T cell test is also not currently approved in either Singapore or Malaysia, and it might also cost a few several hundred US dollars. So therefore, we're probably left with the antibody tests to see whether or not somebody has an immunity. Dr. Leong, if I can turn to you, please, Dr. Leong, I would love to ask you a question about maybe that comes directly from the floor. Actually, it's from the chat group. It's from a gentleman whose name is, a person whose name is Gibson KS Wu. How can cPass be used to assess vaccine efficacy or vaccine efficiency in individuals? What's the threshold and how frequent should we test? Dr. Leong, over to you.
Okay, cPass, reminder, only measures the B cell of it. So we're going to look at one of the two arms. This is looking just on my right arm, and I'm handicapped on the other. So it only measures the antibody levels. And for the Delta, it is well documented, you need 80% and above. For the Omicron, you probably need something more than 80%. We don't have the figure, but if you were to go by 80% or 80+%, I think that's very good. So that's the threshold, 80+%. And how frequent should we do the test? No one really knows, because 30% of the immunity actually stays very well for these people. You can try to do it every 3 to 6 monthly. You can do it every 3 to 6 monthly, and then in turn, you can quantify it coming down, and in turn, you can say, what's your risk like? But Gibson, I think Jensen is going to hate me for saying this, but there's a much better way instead of doing the blood test every 3 to 6 months and say, let's go for the vaccine. The way to do it is actually just go for the booster. Just go for the booster. I know many people in Malaysia have had 2 Sinovacs. Go for 1 Pfizer. And then if a Pfizer opportunity comes along 3 to 6 months later, go for another one and you'll be well covered. If you were to keep waiting and waiting and say, you know what, I don't want to boost until my levels fall to 80%. Well, that's Omicron might just take place before 80%. Number 2, Omicron plus or the next replacement, Yihu or whatever who that comes up could be 90%. So you're kind of waiting. The phrase in Chinese is that you're waiting at the bottom of the tree for a rabbit to kill itself on the tree, and then after that you collect the rabbit. Don't do that. Instead, if I were you, I would just encourage a person, go for the vaccination and see it. Number 2, second reason why you want to do vaccination is not just about antibody levels, but the response of it. After 1 dose of antibody levels, you actually have your antibody levels goes up this way. But after 2 doses, your antibody levels goes up this way. And by your 3rd dose, your antibody protection goes up this way. In fact, you realize with the 3rd shot, your protection against Delta strain, as used in the booster study by Pfizer, becomes effective after 4 days of vaccination, not 14 days, because your body is well trained to it. So it goes back to science. It goes back to what Professor Chan and Dr. Kaur wants us to do: vaccinate, vaccinate, vaccinate. I think it's good to have a test to measure, but measuring doesn't protect you. Getting vaccination protects you. Okay.
Dr. Leong, thank you for your answer. As a short summary, the 80% number that you mentioned for Delta certainly is a very difficult number for us to arrive at. Can I ask a follow-up question, Dr. Leong? When you say 80%, can you explain more about perhaps how cPass, the reports are like, and maybe for a doctor who's seeing a cPass result for the first time, how they could briefly interpret the different percentages? Noting, of course, there's no official guideline on this one.
Okay, so when the first report first came out, they'll tell you whether it's positive or negative. And the initial cutoff they provided was 30%. And the reason for the 30% was this was what we felt was necessary to overcome the original ancestral strain that came out of Wuhan. But as you realize, there was Alpha, and after Alpha, there was Delta. And when Delta came along, the correct figure changed to 80%. So when you see the report, you would have positive because it's more than 30%, or negative is less than 30%. But you cannot rely on that. You need to look at a small print, will tell you the percentage inhibition. It could be 80%, 96%, or 2%. So why 96%? You can imagine one whole plate, one whole slide with many, many receptors that are blocked, where they're lined on the slide. And when you actually throw in the antibodies followed by the spike protein, the antibodies will bind in and prevent these receptors on the slide from showing up. And when it fails to show up, that means you have effective blockade, and it counts for you the number of receptors that were blocked off. So it could be a 96.2, or as we said earlier, as long as you see 80% of the receptors failed to bind to the spike protein, then you have 80% protection, and that's good enough for Delta.
Dr. Leong, thank you. I've received a private question in a chat group. I would like to address this question to Professor Chan, and then maybe ask Dr. Leong about his thoughts as well. Prof. Chan, I'll verbalize the question. A patient has done 2 antibody tests. The first one is a binding antibody and IgG at 27 units per mil. That's the first antibody test and IgG. But the cPass test shows 90%. The question is, does this person need a booster? So the IgG appears lowish. It's an anti-spike RBD, by the way, at 27 units per mil, but cPass shows 90%. I guess an open question to you, firstly, Prof. Chan, and then to Dr. Leong is, what does the doctor do? Prof. Chan, you're on mute, so sorry.
Do we know, we would need to know what type of test is being used for sure for the first test, because we know there are so many so-called neutralization tests in the market, which can actually vary from a rapid kit to ELISA to automated machine to actually something like cPass. So I think it's very important to know exactly if the first assay is actually a rapid kit, then I would think it's not. I would doubt the first results then, because you will not be sure what is it actually measuring, and of course for cPass that is 90%. It really depends, I think, whether it simply means that that's actually neutralization antibody, but to what level, I think I'm yet to see GeneScript to give a data of what that correlates with the WHO neutralization antibody. So I don't have the answer for that. But I have seen that other neutralization antibody tests, such as I think Abbott or Roche, they do provide like any international unit that it's correlates with the WHO SI unit. And however, even with this SI unit, I would like to caution everyone that we are yet to actually find the magic number. So what is the magic number that actually tells you, okay, you are already protected from an infection, or you are already protected from a severe disease? We are yet to actually have the answers for that magic number.
Prof. Chan, thank you very much. Dr. Leong, if I can turn to you for additional
I'm going to
thoughts.
jump in and say that actually that protective level, it's a figment of any person's imagination. That imaginable number could have happened for Delta, but when Omicron changed, the imagined number just changed. When Omicron plus comes along, it will change plus times. So for that reason, I really take pity on WHO. I've been hounding them for a cutoff, but they can't because the virus keeps changing. So I wouldn't count so much on that value, but using an 80% threshold is a reasonable gauge. But coming back to practicality, could we be chasing our own tails repeatedly by choosing a number? I think at the end of the day, it's actually getting vaccinations that's more important. And one of the people which I would really want to do the cPass on are people who are immunocompromised. So how good are they? They might be on mycophenolate, they might be on cyclosporin. I would do the cPass to measure how good they are in their immunity, and in turn can help them prognosticate. You're likely to fall sick, you're not. Your medicine probably made the response worse. Please be extra careful when the vaccine comes along. See if you could take a booster, that kind of thing.
Dr. Leong, thank you. It's very helpful comment and from you and Prof. Chan. There is no magic number to say that above this number you will be protected. And the immune system is very complex, and you will be in all sorts of situations. For example, in an enclosed space, fully ventilated with lots of high viral load individuals. No matter how many neutralizing antibodies that you have, I think that will not be in a protective situation. So you're right that we should probably not wait for an effective number or any number, but rather we could use that as a data point for us to make decisions. This is all very helpful. We have about 7 minutes left, and there will be a very short, say, one slide for Gribbles to tell all the general practitioners about the cPass, the practicalities of cPass. But maybe I'd like to ask Dr. Leong and then Prof. Chan to maybe have any final thoughts. Dr. Leong first, over to you.
I'm just going to respond to Dr. Tan Tin Wee's comment about cPass and sufficient neutralizing antibodies to block. He's right, because this cPass only measures the spike protein antibodies, but you can actually have neutralizing antibodies to other part of the virus, such as the N, for example. But if you were to look and compare which of these are the most important, I can tell you that 90% of the immunity to COVID-19 comes from the spike protein antibodies. And consistently, the spike protein antibody levels are great. Now, there is a narrative that you should use the whole virus for vaccination. That's true. It was stimulated, but it becomes a little bit more complicated in terms of the vaccine, the efficacy, etc. So most people don't. The competitors will be Sinovac, Sinopharm, and Covaxin, as well as a new one which may come into Malaysia and Singapore market called Valneva. But coming back, short of NTN is if you look purely at NTN, you're looking at potentially a 10% window of neutralizing. And given that most of the vaccines from AstraZeneca, Janssen & Janssen, the mRNA, Pfizer, Moderna, everyone targeted NTS, it makes more sense to do a cPass on NTS, neutralizing on NTS, than do NTN as all. And again, the NTNs and the rest play a very small role, 10%, but his point is valid.
Thank you, Dr. Leong, for your final remarks. Maybe I can turn over to Professor Chan for her final remarks about any new points you want to make, or any responses to the other questions or the other comments that Dr. Leong have made, or anything else. Prof. Chan, over to you.
So I also agree with Dr. Leong that we should recommend a booster to your patients. And I think Malaysia is very privileged to actually be able to do I think population-wide booster. I would consider that as a privilege, and with that I believe that we should all take that privilege and do our best to actually.
Thank you, Prof. Chan. You're right about vaccination being a privilege for all of us. And thank you for sharing your thoughts earlier about the science and the microbiology aspects of cPass, and then to Dr. Leong for the clinical interpretations and the uses as well.